Dear friends, today now we are going to discuss about the Kreb cycle. So the previous video presentation clearly explained about the glycolytic process. In the glycolytic process, whatever you consumed during your food intake, the foodstuff converted into glucose. The glucose, 6 carbon glucose is mixed with your blood. So those 6 carbon glucose can be converted into 3 carbon molecule and enter into the Krebs cycle. Okay, so the glucose is travel from one place to another place in the circulatory process, that is called 6 carbon. Those 6 carbon has to enter into the uh, cellular activity with the help of insulin. Because of insulin only, the gate will be opened to enter into the cellular activity. So the 6 carbon has to convert it into 3 carbon molecule. Because of this function, we need to secrete uh, optimal level of pancreatic secretion. Because of pancreatic only, the the glucose can be entered into the cellular activity. So after converting 3 carbon molecule, the 3 carbon has to convert again into 2 carbon acetyl CoA. So with 2 carbon acetyl CoA only, it is possible to observe your mitochondrial reaction. So then only the mitochondrial reaction will be perfect. Otherwise, it will be get collapse. This is called metabolic. If there is any problem in this process, it will be called as a metabolic disorder. Okay, so if the process is continuous, there will be no problem. The excess glucose can be converted. The excess energy reservoir can be uh, diluted and expelled through chemical and heat energy from your body. Let us discuss one by one. So Krebs cycle is nothing but it is a serious enzymatic reaction that take place in the mitochondrial, resulting of formation of acetyl CoA to release carbon dioxide, hydrogen, and later on formation of water. This complete process we used to call it as a Krebs cycle. So here there is a question: why it is called Krebs cycle? Krebs cycle is nothing but it's a name of the person who is invented this process right from 19th century to 1981 to okay, the person who is spending his whole tenure of his life and um isolating and invent this process. This is happen in the cellular activity. That's why we call it as we fix the name of scientist for this process, that is called the Sir Hans Adolf Krebs cycle. Actually, the scientist full name is Sir Hans Adolf Krebs. Krebs cycle also called other names are citric acid cycle, TC cycle, or Krebs cycle. In this way, you can call this process, but universally accepted Krebs cycle. So, in this Krebs cycle, pyruvate formed at the end of glycolysis. So, pyruvate is nothing but 3 carbon molecule, uh, which will enter into acetyl CoA for formation of oxidative glycolytic process. Okay, so the citric acid cycle is final common pathway for oxidation for all biomolecules, and this will happen. See, acetyl CoA can enter into the Krebs cycle, but acetyl-CoA, it is not because of not only the carbohydrate, but also it will derivate from protein and fatty acids. So all together, the protein, fatty acid, carbohydrate has converted into 2 carbon molecule and finally enter into the mitochondrial reaction or Krebs cycle. In this process, we need 8 coenzyme. Because of these coenzyme only, it is mediated the chemical reaction and form the entire process. So in this, at the end of this phase, it will enter into the electron transport chain and make your 12 ATP into the multiply into 3, 12 into 3, so 46. It will happen at the end of electron transport chain. So as we discussed earlier, the acetyl CoA is not acetyl CoA is formed from carbohydrate, protein, and fat. So glycogen is converted into glucose, pyruvate, and finally enter into the acetyl CoA. For this process, glycolysis has to create has to initiate. And second process is triglycerides lipolysis process, and beta oxidation finally enter into the acetyl CoA. And the protein converted into amino acid enter into the acetyl CoA with proteolysis. All this process finally enter into the acetyl CoA in the Krebs cycle. So first step of Krebs cycle is very simple: decarboxylation of pyruvate into acetyl CoA. So actually this is first phase. 3 carbon pyruvate converted into 2 carbon acetyl CoA. So the entire process, the conversion of 3 carbon into 2 carbon molecule. So pyruvate, it consists of 3 carbon. It has elevation of 1 carbon from its pyruvate, and releasing of 1 carbon, and form as a acetyl CoA with the 2 carbon. This 2 carbon only it will enter into the Krebs cycle. So if there is any problem in this conversion, there will be no possible of citric acid cycle functioning. So actually, this is the this is a pre formation of the condensation of acetyl CoA with the oxalate. So this is actually first phase. 2 carbon has to combine with 4 carbon oxalate and formation of 6 carbon. So acetyl CoA, it contains 2 carbon. Oxaloacetate, it contains 4 carbon. These 2 and 4 combine and make it as a 6 carbon. So acetyl CoA, oxaloacetate combine together and make it as a citrate. So in this format, acetyl CoA has to include already it contains acetyl CoA. This acetyl CoA has to remove, and this carbon, 2 carbon in the chain, and make it as a 6 carbon. So this still the carbon molecule chain, it is changed here. You can see the first steps citric acid cycle is join 4 carbon compound with oxoacetate of 2 carbon compound. So totally it is a 6 carbon molecule. Citric acid cycle first phase is actually generated, and resulting of citric CoA, which is later cleavated free coenzyme of A, form citrate. So it is very simple process. Oxaloacetate, it contains 4 carbon, and acetyl CoA contains 2 carbon. These 2 and 4 make it as a 6, that is called a citrate. So in this process, one hydrogen has to include, and the succinyl CoA, and that succinyl CoA has to removed from this process so that the oxaloacetate can be converted into citrate. This is first. Second stage is isomerization. Isomerization means there is no change in that portion, but one dehydration, one rehydration. Here we are taking out one hydration, here inclusion of one hydration. So the chemical bond will be differ from citrate to isocitrate. Okay, here one hydrogen is removed from this chemical compound, and here one more one more carbon dioxide, water molecules inclusion. So the chemical bond will be differ from citrate. Okay, in this phase, this HOH, it will be converted as a water molecule. And again, here the total position is changed. Because of this change, we have to go for dehydration and rehydration and the second phase with the help of acuminase. So because of this acuminase, the citrate is converted into isocitrate. So isocitrate converted into uh, citrate is converted into isocitrate through formation of intermediate cis acuminase. Okay. So the reaction is reversible reaction catalyzed by acuminase enzyme, and this process is taken to process dehydration of forming of cis acuminase. Second step is rehydration cis acuminase into isocitrate. Okay, the exclusion of one water molecule, inclusion of one water molecule. On this process, this is called isomerization of citrate into isocitrate. Okay, third phase is very simple. There are two processes: dehydrogenation and decarboxylation. Dehydration means we are going to remove one hydrogen and carbon dioxide. So let us discuss. So 5 carbon compound into 4 carbon compound. Okay. So in this process, we are removing one hydrogen, dehydrogen hydrogen, and here removing two carbon the carbon dioxide. So this is called iso, with the help of isocitrate dehydrogenase only. This the formation of isocitrate into ketoglutarate. Okay. So third step citric acid cycle is first of 4 oxidation reduction. Again, the 6 carbon molecule into 4 carbon molecule enter in this ketoglutarate. Okay. So isocitrate oxidatively decarboxylated from 5 carbon compound into ketoglutarate catalyzed by isocitrate dehydrogenase, and this reaction is like second reaction, two-step reaction. First one is isocitrate is dehydrogenated to oxalosuccinate, and the second step is decarboxylation, oxalosuccinate. This is oxalosuccinate. The first step result in formation of NADH, and the second step involved carbon dioxide removal. Okay, so this is about third stage. And fourth stage, very simple. The carbon dioxide, again it is decarboxylation. Here you can see the word decarboxylation. We are going to remove and from one carbon dioxide from ketoglutarate. So this will be converted as a succinate OA. So 5 carbon into 4 carbon molecule again. Okay, so reaction irreversible catalyzed by enzyme ketoglutarate dehydrogenase. Ketoglutarate dehydrogenase. So because of the removal of carbon dioxide, we need ketoglutarate dehydrogenase complex, so that the ketoglutarate converted into succinyl CoA because of decarboxylation. This reaction is similar to oxidative decarboxylation of pyruvate involving reduction of NAD plus into NADH. So fifth stage, conversion of succinyl CoA into succinate with help of succinyl CoA synthetase. In this process, guanine diphosphate converted into guanine triphosphate. From succinyl CoA, it will remove one CoA, so that it will be converted from succinyl CoA into succinate. So there is a, if you remove this, the chemical bond will be different from succinyl CoA. So In this step, succinyl-CoA undergoes the energy conserving reaction in which succinyl-CoA is cleavated from succinate. Okay, this reaction accompanied by phosphorylation, guanine triphosphate converted into guanine triphosphate because of one phosphate is releasing from this succinyl-CoA. Okay, so guanine triphosphate is converted into guanine triphosphate is forming two ATP molecule in this process. So one guanine triphosphate is generated and two ATP molecule is formed. Sixth phase is very simple. Again, we are going to remove one hydrogen from this succinate. So if you remove one hydrogen, two hydrogen from this phase, succinate may be converted into fumarate. Okay, this will happen in the intramitochondrial space with help of succinate dehydrogenate. Okay, so in this phase, as we discussed earlier, two hydrogen is removed from succinate. So succinate can be converted as a fumarate. Okay, this will happen in the intermitochondrial space, and in this chemical reaction, at the end we can get FAD and FAD. FAD வந்து FADH convert out. So FADH then enter into electron transport chain via complex 2 and finally forming of 2 ATP. The seventh phase, hydration of fumarate into maltate. We are going to inclusion of one more hydration in this process. So fumarate may be converted into L-malate. For this process, one hydrogen is inclusion. So again, one more hydrogen is included. So H2 is included in this phase to forming a L-malate. So eighth phase is very simple. Again, we are going to remove two more hydrogen from its from L-malate. So because of removal of two hydrogen molecule from malate, it will be converted as a oxaloacetate. The oxaloacetate, it contains of 4 carbon molecule. So at the end of eighth step, the Krebs cycle is going to start one more cycle. So in this process, L-malate is converted into oxaloacetate. For this process, NAD plus converted into NADH. So this reaction complete the Krebs cycle. So in this way, each and every time the Krebs cycle runs the conversion, hydration, dehydration, and decarboxylation. Because of this process, it starts from the 11th stage to 18th stage. For this stage, 8 coenzymes are involved. Finally, you will get oxaloacetate from malate. Okay, as we discussed, the pyruvate, the glucose converted into 3 carbon molecule into two different pyruvate. So these 3 carbon molecule cannot be accepted Krebs cycle. So again, it has to convert into 2 carbon molecule as acetyl-CoA. This acetyl-CoA only enter into citrate. For this process, this oxaloacetate has to convert with acetyl-CoA. So oxaloacetate, it contains of 4 carbon molecule, and these 2 carbon molecule make it as a 6. And the third stage, it will be removed. So one carbon will be removed, and the fourth stage, again one more carbon removed, and remaining phase it will travel 4 carbon molecule. In this phase, there are different enzyme, different names of the phase. So it has to travel from one stage to another stage by inclusion of our oxygen, water molecule, and exclusion of water molecule, and inclusion of carboxylation and exclusion of decarboxylation from its chemical bond. So Because of this reason, citric acid cycle is run every minute. So because of Krebs, the Adolf Kreb only, we are able to recognize your chemical reaction within our body in the mitochondrial reaction. If there is any problem in the mitochondrial reaction, definitely there will be a problem of metabolic disorder. Because of metabolic disorder, you get overweight or low weight, or maybe high concentration of sugar present in the body. Because of Krebs cycle reaction only, the excess glucose can be converted and transferred from blood tubes to into cellular activity with help of insulin. Because of insulin only, the 6 carbon divided into 3 carbon pyruvate. These 3 carbon pyruvate again converted into 2 carbon acetyl-CoA. These 2 carbon acetyl-CoA combined with oxaloacetate and make it as a citrate. The finally you will get oxaloacetate. Okay, thank you.